Oh, hi everyone. Welcome to the IBH webinar series. I'm Yunqing from the James Hutton Institute, and today we have the pleasure to invite Dr. Chen Ji from JIC. And Dr. Chen Ji, she obtained her PhD from Chinese Academy of Sciences Center for Excellence in Molecular Plant Sciences and Institute of Plant Physiology and Ecology in Shanghai, China, where she studied the development of maize endosperm. And right now, she's a postdoctoral researcher in the lab of Philippa Boreas Group at the John Innes Center. Her research focuses on analyzing the spatial organization and transcriptional activation of genes in wheat grains at different stages after flowering, using techniques such as single nucleus analysis and also spatial transcriptomics. She's particularly interested in the genes regulating the nutrient and micronutrient uptake and accumulation. The ultimate goal of her research is to enhance the nutritional value of wheat and contribute to address the hunger and hidden hunger. With that, we welcome Dr. Ji for today's talk.
Yeah, thank you, Yunqing. Thank you for the introduction. And just let me to share my slide first. Maybe not here. Okay. Sure. Is that okay with my slide? Is everything okay?
Looks great. Thanks.
Cool. Thank you. And I can start. Hello, everyone. I'm Chen Ji from Philippa Boreas Lab, joined the center. I'm a postdoc for now. And today I will talk about a new method, but actually it's not that new, and it's called the spatial transcriptome. And I just use this method and to analyze the wheat grain and to find a lot of the gene expression very specifically, and also try to find some of the genes, like for example, some of the sucrose transporters, and to find their gene function. First, I want to introduce for the wheat, maybe a little bit on the barley, because this is the pictures I took last year in Australia, and because when I get into the shuttle bus, because I want to go to the field station, and when I was in the shuttle bus, I was like, oh, there are a lot of weeds. But when I get into the near to the plants, and it turned to the barley, I was like, oh, it's quite similar, because wheat and the barley, I guess they are very close relatives, and they share a lot of the conserved genes, and also very similar the grain structures. And also many gene biological pathways are quite comparable, and also between these two species. But I have to say, I know that barley also have a very big genome, but for wheat, it's more hard to analysis because it is the heteroploid. It has the A, B, and D. That means if you want to clone a gene, actually you just clone the 3 of genes, and also the A, B, and D genes share the similarity to 98 to 99% percentage of the similarities, which is very hard to know the gene functions. But the people are still trying to use a lot of the method to know the gene function. For example, they try to using the different method to get the RNA and to know the gene expression pattern. And for the gene expression pattern, we already know that some of the website we can check the gene expression pattern. For example, if you're working on the apoptosis, that will be very easy to get the gene expression pattern, by this website TIRE or the ePlant. You can see the gene expression pattern by the EFP, and you can see some of the genes. This gene is highly expressed in the leaf, in the some kind of a stage of the leaves. And for barley, I know that you already built a very beautiful in the website, and you can check the gene expression pattern by this website by the different tissues, and also by the different environment. For example, the cold tolerance or the heat stress. And for example, this gene is highly expressed in here. Maybe I should using the laser pointer. It's very highly expressed. It's you can just straight to the point and to see what is kind of tissue, and also to see what kind of the environment they involved. And for our wheat, and it's also have another website, and you can check the details of the expression patterns. And for the wheat, I want to introduce more about the expression pattern. because at the beginning it's very hard to get the genome, and also it's very hard to get like the RNA-seq data. And for the several years of trial and tried, and scientists are trying using the different tissues and to build expression pattern atlas with the bulk RNA-seq data. For example, I'm working on the grain. If I want to find something just to test if my genes express in the grain, I just using this kind of method. I using the bulk RNA-seq to get the grain and extract the RNA to send the RNA-seq to test if the gene is expressed in the grain or not. But if we want to see the details of the grains, and actually the grain can divide into roughly divided into 3 parts. And the outlier is the pericarp. It's very easy to peel, and you can just use the knife or the tweezers and just peel off the pericarp. And the inside is the embryo and endosperm. And you can just using the dissections and to dissect them and to do the RNA-seq and to get the RNA-seq data. And also, if you want to see the more details of the grain, for example, the endosperm, and you can using for example, the micro dissections or the laser capture micro dissections, and to see the details of the grains. For example, this is the picture of the endosperm. If you want to see the endosperm very detailed, and you can just put the endosperm into the microscopy. And the outer layer of the endosperm, they're kind of like cells, very small, very dense. It's the different from the other cells. The other cells are round, but the outer layer of the cells is in squares, and it calls the elytron layer. And also it's same with the transfer cells. When you dye in with the paraffin sections, the transfer cells are different, their colors are different. And the rest of them, and they just named with the starchy endosperm because it's full of starch. And also you can use the laser capture micro dissection. You can just find very carefully to dissect one layer or the several layers, and after that you get you punch of cells and you do the RNA-seq. It's very useful to see the for example, the elytron layer, to see their gene expression pattern in the elytron layer. And for nowadays, there are lots of the technique. And for the single cell sequencing and single nuclear sequencing, I guess a lot the people already knew a lot of the papers, a lot of the technique for the single cell or the single nuclear sequencing. It's very useful. It gives us the single cell or the single nuclear resolution of our for the different tissues. But for the wheat grain, it's very hard to use both of these two the technique. For the single cell, both of them need, for example, both of them, they need to use the enzyme to digest your cells, especially for the single cell one. But for our samples, the pericarp, it's very hard to digest. But for the endosperm, it's very easy to digest. You need to balance them, for example, they need to be in the same environment, the same temperature, the same time, and it's very hard to. Sometimes you just get a little bit of the pericarp. For the endosperm, you just get a lot of the endosperm. It's not fair for the endosperm, right? Or the single nuclear sequencing, it can work for our tissues, but our tissues still have some of that remain solving problems. For example, we have a lot of starch, we have a lot of proteins, and the starch size is sometimes the starch size is the same size with the single nuclear. You're just very hard to separate the starch with the single nuclear. And also, if you use the single cell sequencing or the single nuclear sequencing, it's just you get a lot of the single cell, the single nuclear. You just sequence the cell by cell and the nuclear by nuclear. After that, you need to remap them with the marker gene. Marker gene is kind of genes that only you already know their expression pattern. For example, this marker gene is you already know that this gene is expressed in here. And when you do the remapping, you can use this kind of marker genes. But for our grains, it's very hard to get the marker genes. It's easy to get the pericarp. It's easy to get the modified elytron because sometimes you just using the morphologies and using the microscopy and to dissect them and to know the gene expression. But for the especially the starchy endosperm, it's very hard to remap them because it's just a bunch of cells. You can't tell the differences between with the morphologies. So we just stuck a little bit for the single cell sequencing or the single nuclear sequencing, but during the 2020, the spatial transcriptome has been selected as the method of the year. And it's just the beginning using for the human tissue or the animal tissues. We're thinking that maybe we should give a try on this method. So if we want to doing the spatial transcriptome, the first stage is to select your stage. This is, I just quickly scan our samples with the microscopy, with the paraffin sections to see the cell division, cell differentiation, and also the green filling, and to see the starch of the proteins. What is the time for the starch getting into our grains? And for the grain development, it can divide into several parts, 2 or 3 parts. So the first part is the early development. There will be a lot of the process involved. Include with for example, nuclear divisions, and also cellulizations, and also a lot of cell differentiations, and also a lot of cell divisions. And after the early development, there will be grain filling stage. Grain filling, that means it's time for grain to store a lot of energy, and the sugar is just transferred from the maternal tissue and to the grain. And in that stage, I was thinking that maybe it's a good stage for us to analysis, because there are lots of working on the grain filling, and the grain filling is very, very important for the grain for the wheat analysis. So we just picked the 12 days after the flowering, and also during the grain filling stage, they will be coupled with the mature stage, and during the 80 days and to the 20 days, and they will be start with the programmed cell death. And that means if the grain start with the programmed cell death, especially in the sperm, and you will lose a lot of RNA, because the spatial transcriptome is the method that you will collect the RNA. But for the PCD, you will lose of RNA. I was thinking maybe the second stage will be the middle stage of that, and that will give us more of the RNA, but not involving too much of the programmed cell death. So we're thinking take these 2 steps. Also, because the Candida is a kind of a wheat varieties, it's widely used in the UK. So we're just using the Candida Y type, and to have the 12 day after flowering and 80 day after flowerings. And after we select our stage, and we are trying to do the real spatial transcriptome. And at the first step is to do the embeddings, because we're trying to make the most using our chips, and we just try to have the different type of the sections. For example, for here we have 2 of the cross sections. One is just cut into the middle of the grains, and one just cut into the middle of the embryo. That means we can have all the cross section of the sperm, and also we can have the details about the embryo, and also we will have the longitudinal sections, of course. After we just set on doing our embedding, and after doing the cross sections, I just put the other to do the spatial transcriptome. And after we do the frozen sections, we just have a very thin of the sections, and put our sections into a chip. The chip actually it can divide into 2 parts, and the first part of the chip it has a very tiny spot, and the spot is around 0.5 micrometers. It's very small. And after that, you just using a lot of enzyme to digest your tissues, and your tissues will release the RNA, and the RNA can drop into that tiny little spot. And in each tiny spot, it has the beads. That is the second layer. The beads has a tag with the address, and also the beads can capture the RNA. And then the rest of the method, the analysis, the rest of the experiment will be very similar with the bulk analysis. You just do the library, you just do the sequencing, and you can map your data. But for the mapping, that will be a little bit different, because normally with mapping, we just mapping with our transcription the data or something. But for our mapping, actually, it's not only the transcript data. It's also have a little tag of the address data. The address can let you to remap your samples into, for example, this spot is come from here, and you can just remap it in here. And that is after that, that will be the visualization. And after that, you can see the details of your gene expression pattern. Okay. And this is some of our data, and because we have like 3 replicates, and I just picked one of the replicates to show the data, and we can see the left hand is the DAPI staining of our samples. There are two of the stage. One is 12-day after flowering, and one is the 8-day after flowering. Each of them has the one of the cross sections, and one is a cross section with the embryo and also the longitudinal section. This is like the detail of our chip. And after we get our samples, it's like we just try to a little bit analysis our samples. And this is a heat map of this chip. And the red means in this area, there will be a lot of the RNA reads. And we can see in the embryo, there are lots of RNA in the embryo, and also it's the same with the outer layer of the endosperm. Okay. But here now, I just have the data and I need to analysis. But before that, maybe I should refresh with the green, the green structure. This is a green, and the out of the green, and here, this is a pericarp, and inside the green, and it can divide into the embryo and endosperm. For the endosperm, the out layer of the endosperm, the tiny little squares, and they're called the aileron layer. And also it has this is a vascular bundle. It's just a bunch of cells can get it with can link the grain and to the spike. And also is above the vascular bundle, there will be modified aileron. It's very similar with the aileron, but their morphology will be a little bit different. After we get our samples and we analysis and we cluster them, we will have this this is one of our samples. And they will give us the picture like that. It's called the spatial cluster spot. And after that, how we can analysis this data, because this kind of the spot plot is just based on the similarity in the gene expression, the profile. If we want to read about this picture, we can see, for example, the out layer is very purple. It's called the aileron layer, and their expression pattern are quite different with the rest of the cells. And also in the middle, there's a vascular bundle. That means if you want to select some of the transporter, the Sucrose transporters, amino acid transporters, you probably will select, they will highly express in here, because here it's just a link to the modified, it's just a link to the spike and to the grain. And after they transport something from the vascular bundle, and they will spread out through the modified aileron, and also quickly transferred into the a different kind of the endosperm. Because this is only the one slide of our samples, we have 6 slides in total, so there will be 12 clusters. Okay, how can we use these clusters? And first of all, we probably will using some of the clusters and to analyze some of the unknown genes. For example, this is a heat map that I selected some of the genes highly expressed in the different clusters. And I just randomly select a gene this is a gene from the cluster 10. And cluster 10, we can see that, the cluster is highly expressed in the here. That means the aileron of the 12-day after aileron. And in the NCBI or the ensemble plants, if we just select these genes, we just the annotation will be unknown protein. We know that if you just blast the protein, we probably will have the annotation of this protein, but not that accurate. But we can combine our expression pattern this one, and to predict our gene expression, gene function. This gene must be involved in for example, the modify the aileron the function, right? And also there we still can have to see the gene expression with our already known genes. For example, we have a lot of sweet gene is a kind of gene family that has a lot of genes. It has, I think it's more than 100 genes. But actually, even the different sweet genes, there are a lot of the sweet genes expressed in the grain. If we want to see the details of the grain, the gene expression pattern, we can just simply using the gene ID with the sweet genes. For example, this gene is highly expressed in the EAS, because EAS is kind of the area can transform is kind of the cells between the embryo and endosperm. That means this kind of gene maybe can transfer from the Sucrose from the endosperm to embryo, or the other way around. And also there's another there is another sweet genes expressed in here. When I searching this gene, I was thinking, maybe this gene is not highly expressed, but it's just reported, but it's highly expressed in the green. Then I just noticed that in here and here and here, this is a vascular bundle, and this is bright white, because our wheat has the A, B, and D. If you want to see the the gene expression pattern, it's okay, because in here, the B is the A, green is the A, and the purple and the green and the orange will be the different the A, B, and D the gene. But for this gene, because in each spot is all A, B, and D all expressed, and that will be bright. That means this gene is very specific expressed in the vascular bundle. That means that this kind of sweet genes must involve into the maternal and to the grain, and they're the sucrose the transport this kind of function. Besides the sweet genes, I also check some of the other the transporters. I also check the amino acid transporters and the other the transporters, and also I just found a very interesting gene. This is one of the genes called the SOT1. When I check in the genes, this gene is very highly expressed compared to the other transporters. It's just the 30 times or the 50 times. It's very high, and also their expression pattern is very different. It's expressed highly on the out layer of the grain, and also is the editing layer, and also is highly expressed in the modified editing, and also is highly expressed in the out layer of the end-of-sperm embryo. That means this gene must involve in the different parts of the tissue sucrose interaction, right? So I want to make sure that if we have more of the SOT genes also involved in this pathway. So I just checked all the SOT gene in wheat, and the SOT1 is the gene that only this gene highly expressed in the grain and expressed that specific. So I was thinking, maybe it's time to have the functional validate. So Emily and James just helped me to do the crossings, because I'm using the cadenza wild type. The cadenza has the A, B, and D. If I need to have the triple mutant, that will cost around a long time to go. But I'm very lucky and to search the tele mutant, and it has the chrono background the mutant. Because compared to the cadenza, it has the A, B, and D. The chrono only has the A and B. That means that if I just select this kind of the mutant, that can help me to save some time. And also I can do some of the background backcross. So they just help me to using the chrono and to do the crossings. And yes, and for at the beginning, I was trying to find out the phenotype, but I was thinking that we must have the double mutant. The double mutant, the gene expression is so strong, it must have the phenotype. But when I screening all of my plants, it's very hard to find the phenotype. It seems all like that, and but after do the genotyping, I just noticed that we only have the Y type and the A mutant, B mutant, or Het mutant, but we really can't make the double mutant. But after we tried, and we just plant around 200 and more of the plants, and we just get some of them very small, very weak, very unhappy the double mutant. Or after we do the genotyping and we know, this is a double mutant, we can't have any seeds from the double mutant because it's too weak. So if we want to see the detail of the green phenotype, because that one is highly expressed in green, it must have some the phenotype, I guess. So we were just using the Het A mut B or the mut A Het B to find out the phenotype. This is one of the spike. This is one of the spike. If you want to see the detail of the spike, you can see that some of the grains are different from the other grains. It should be a little bit about pale and also shriveled. Okay, it's here. And we just using this kind of grain and to do the genotyping, and we just noticed, this is the double mutant, and most of them can't generate a very big plant. And also for the double mutant, I try to figure out what is the detail of the function, what is the detail of the phenotype. So I just do the preference sections of the A mutant, B mutant, and also the double mutant. It's very similar with our phenotype for the plant. It seems like the A mutant and B mutant, there is not a big differences compared to the Y type. But for the double mutant, when I do the embedding, and my seeds just floating, it's very hard to do the embedding. And when I do the cutting, I just notice, oh, it seems like there is a big hole inside. So I just want to see the details of our samples. And we can see that in here and here, because the SAT1 is highly expressed in the modified aileron and ailerons. We can see that it seems like the morphologies for the modified aileron, it seems like there is not a big differences. Maybe the SAT1 is not involved in the, for example, cell lysations, the cell differentiations, or the cell divisions. But in here, maybe they just try to do something. And because SAT1 is kind of like a sugarous transporters, it must be the transport the sugars, right? So thinking that maybe here, there will be something in here, maybe some water, maybe some solution, maybe something. I really want to see that if the sugars will be the differences, will be a big differences compared to the Y type and the double mutant. So we're trying to use the MALDI and to analysis what's inside of the double mutant compared to the Y type. So the MALDI is very similar. At the beginning, the MALDI is very similar with the spatial transcriptome. We just embedding our samples to do the frozen sections. And after that, you just handle your samples into the slide and put your slide into the MALDI the machine and spray the solution and using the laser to point spot by spot. And after you get one of the spot, or several of the spots, you can analyze your data with the MS. Because we were using the non-target of MS, it's more like or you will get everything from that spot. Because I'm working on it must it might be differences between the sugars. So I try to figure out the sugars. The sugars, the MZ ratio is around 365. So I just pick this peak. And after that, to do the visualizations. What I have done is, what I've got is for the Y type and the mutant, there will be a big differences. We can start with the Y type, and this is the vascular bundle, it looks like that. And this is the modified aileron, it looks like that. And besides that, this is called the cavity. Most of the sugars are gathered into the cavity and transport by maybe the SAT1, maybe the other sugar transporters from the cavity and to the sperm. But for our SAT1 mutant, it seems like the big hole here, their gap here, is just full of the sugars. And the sugars, because we are lack of the SAT1, it can't transport from the cavity and to the endosperm. That is why in our endosperm, when we do the sections, there are less of starch, less of protein, and also there is a big hole in the hair because it's full of starch. Okay, this is only the one example for me, to using the spatial transcriptome to find out the gene function. Actually, in wheat, there are tons of genes, and a lot of the people around the world is working on the genes, working on the wheat genes. So we were, with the help of the informatics platform in JIC, they're trying to help me to build a website that can let the others can also using our spatial transcriptome data to find more of the gene expression pattern. And called the Gene X, and unfortunately it's not released for now because we are still update our data, but I think it will be released in December. Okay, this is the website of the website of the spatial transcription data. And if we just click into this website, and you can see here the page like that, and it has the different data set. It has the 3 of our replicates, replicates 1, 2, and 3. And also it has the BN50 and BN100. What is the BN50 and what is the BN100? Because when I analyze our data, because we're begin with the BN1. BN1 is, that means the 0.5 micrometers. But actually BN1 has a huge matrix. It's very hard to analysis, and it's very hard to finalize what is the gene expression pattern because the spot is too small and it can't be visualized very well. So we just have the BN50 and BN100 resolution. BN50 will be 25 micrometers and BN100 will be 50 micrometers. and you can choose whatever like the replicates you like and whatever like the resolution you like. For example, I just choose the replicate 3 for the B50 resolution. That will be 25 microliters, and the page will be like that. In the middle is our like the sections, and this is the depth staining of our sections. If you just simply like the type the Chinese the gene ID with the Chinese Spring version 1.1 in here, and just type here, and you can see the gene expression pattern like that. This is a SAT1 genes. I know that I already seen the like a SAT1 genes. It's very like similar with the expression pattern compared to this one. Okay, I think is like this is the using of our website, and I think that is most of my work. And to generate, I just using high resolution spatial transcriptomics data and to analysis the weight grains. And also I just identified transporters, like for example, Sucrose transporters, and to try to analysis of also like genes. And also we will provide online database that can allow the people out of the JSC, out of our lab to analysis the gene expression pattern with the weight grain. Because our website is still not available, but I think I will be keep updating about our samples. And in the Blue Sky, if you want to take a look on that, please follow me on the Blue Sky. Okay, and last, I want to like thank all of my lab members, especially like Philippa. I just waste a lot of money. Thank you. And also like Annie just had me to do a lot of crossing, and also Holly Sam. And also there is a big help with within the GIC, for example, the proteomics, informatics team, whole group culture team, and the bioimaging team. And also there a big thank for Alison, David, and Guy, and for the multi-samples analysis and multi-sample to do the multi-samples, and Katie and Crystal to chat about the stereo-seq and the other stuff. And thank you all for the listening, and I'm very happy to have any questions. Yeah. Sorry, I can't hear you.
Yeah, now should be fine. Sorry, I was
Yeah.
muted. Yeah, thanks, Chen, for the very nice story and very powerful technique. And I see there are already some questions from the chat box.
Yeah.
And the first one, this is from Rinxuan Zhang. And I don't know if Rinxuan want to ask him his own question. I think I can just read it here. He wondered, how does the stereo-seq compare to the Wheaton system from 10x?
Yeah, actually, because like before I joined the Barley lab, I was working on the 10x1 because I was working on the maze, and also I involved in the project working on the maze spatial transcriptome, also in the grain, but using the different platform using the 10x genomics. The big difference compared to the 10x is the stereo-seq has the higher resolution, because for the 10x, I know that the update, I think they update their like the technology or something. But for the plant, we still can only use the resolution, only have the 55 micrometers. But for the stereo-seq, if you want to see the very detail of your samples, there will be less than 0.5 microliters. But you can analysis your data with the Benton, that will be the 5 microliters. It's around the 5 micrometers is around for example, aerial layer or the embryo or the root or some of the leaf samples. It's the same size of the single nuclear. So in my theory, I think the stereo-seq will give you the more of the higher resolution compared to the 10x with them.
Yeah. Sorry, I have a follow-up question on that, and I just wonder what is the accuracy of this, because I remember Cristobal, he gave a talk at some point, and he said he has tried the stereo-seq some years ago and didn't work that well for him, but he also said that it was a version several years before. So I just wonder if you can give some comments about that.
Yeah, because actually our the project is just the same start with the Crystal. So we just chat a bit. That is why I just leave the thanks at the end of our our seminar. because they're working on the spike, I guess, right? Because the spike cell, they're pretty small. And when you do the seq or all the spatial transcriptome, you need to be very, very clear, like very careful about your samples. If you want to cut a slide, that's okay. After you cut your slide, you just need to move your slide into the chip. That's okay. And after you do the optimization, that means that you have a lot of like the enzyme to digest. That means this is your samples and this is the chip. Actually, they are stick to each other. But when you put your like a solution here, there will be some if you're not very careful or something, and there will be for example, this is a spot and this is a spot. This is your tissue, and there will be some of your RNA will release to the other spot. And this is comes to like I think I can show you some of our data here, because in our data you still have a little bit for this one, but not too much. We can see the heat map. What's the heat? Oh, here. Actually, we can see that in here. This is not that good. Like for example, when you do the like the permeabilization, you just move a little bit, and there is some of the RNA release that. You can analysis a little bit. For example, you can like get the background out of the here, but it's still very hard to do that. In this step, you need to be very, very careful, especially for like their samples are like the spike or the root, and their cells very tiny, so it's very hard for them to do that.
Yeah, that's good to know. And there's another question from Wen Hao. Do you think the huge gap of SUT double mutant is the bigger endosperm cavity?
Yes, actually, we want to. Okay, all of animations. Okay, here, I guess so, because like this is not a perfect like the preference sections. We can't see like the detail of the like the modified aileron, but we can see that this is a modified aileron, this is aileron, and sometimes this is all the endosperm, right? And for here, I think it must be the cavity, but the cavity maybe like is too much because it's full of like the sucrose. It can't be transported here, because if the sucrose just stuck here, the concentrate will be very, very high, and they will be stuck with the water. So the cavity will be bigger and bigger and bigger. That means I guess that will be the bigger of the cavity.
Okay, thank you. There are other questions from Chandler, and he said, or she, thank you for the nice talk. Did you try cell segmentation using DAPI-stained images for cell-specific expression?
Yeah, that is a very good question. Yes, I tried a little bit, but not too much, because for our samples, I didn't show here. I think the cell segment is not working very well for our grains, because when I doing the DAPI staining, normally because they're like the protocol is working on like the other tissues, and they're working, I think it's okay with the cell segment with the DAPI. But for our samples, when I do the DAPI staining, and some of the starch also died a little bit. So it's very hard to tell the differences between the nuclear and the starch. And the machine, like I know that I can use the machine learning, but for the time for now, I just to move forward and to analyze our data with the bins. But it's very good to have if I can have the cell segment with the DAPI, that would be very perfect, because our for the grains, they're like a cell type are so different. In the middle of grains, they're like a very big cell, and the out of grains are very small cells. If I can do the cell segment, that means that we will have like more of the data for our like the single cell. But till now, I think I'm just moving on with the bin analysis, not the cell segment analysis. But I will try later, I think.
Yeah. I have a follow-up question on that. So you mentioned that your data kind of divided into 12 clusters.
Yes.
And it's based on different type of cells. And this is based on your knowledge of the cell type, or is based on the expression data that they really just automatically group into those?
Yeah, that is a very good question. If you want to analysis with the cluster or something, it will be a lot like the things that you can do. If you want to have more and more clusters, you can have more and more. For example, I'm just for example, the cluster that I show with the other with this I use like the bin 100, I think, and but I will analysis with the bin 50 or the bin 25. But actually, if you have the bin 50 or the bin 25, there will be more and more clusters. That means that you will have, of course, you have like the more of the like the smaller cells, and you will have the more detailed cells, and you can have the more and more clusters. But you have to like the balance of the, for example, the explanation. Also, if you want more, and you can like the analysis more. For me, for now, I'm happy with like the 12 clusters, but I'm trying to get rid of the endosperm to only the using the embryo, because the embryo, the cell is like the more like the more average and the more small. I try to using the bin 55, bin 25 analysis, and they will give us like the more and more about the subclusters.
I see. Okay, so there's another question from Chandler. The question is, what was the transcript capturing efficiency for your samples per micrometer or 10 micrometers?
Sorry, for the 10 micrometers or what is the micrometers?
Yeah,
sorry.
So the person asked the transcript capturing efficiency for your samples, every micrometer or every 10 micrometers?
Actually, we don't calculate like that. We call like the bin, like for example, like the bin 1, that will be something, like the bin 1 will be 0.5 micrometers, I think. But bin 1 is very, very tiny, very, very small. And if I want to like to test the capture accuracy, that will be like the bin 10 or bin 50. I'm not 100% sure when I do the analysis, but it definitely will be something like that. Yeah.
Okay. I don't see other questions in the chat box right now. Maybe I can ask another question. So you mentioned that for your double mutant, your SUC double mutant that didn't really survive. And this is because the endosperm was not developed properly.
Yeah.
So if you grow your seeds on some agar medium that you supply extra sucrose, would you recover the plant and could it grow normally, or there's still some phenotype?
Yes, thank you, because we tried. We tried a little bit more, but not too much, because we tried to separate the embryo. Because at the beginning, I was thinking that, oh, the SAT1 maybe have the very like a strong phenotype on the like the endosperm, on the aileron layer. So maybe the embryo still can survive, right? And we can have the like the double mutant. It must be like the some kind of like the embryo. So we tried a little bit, but the result is not so good. So I don't think that we tried a lot, but none of them survived. So we're just thinking that maybe we just keep this like a double mutant.
So do you know if this SUC1 gene is very specific to the seed, or it's actually also expressed in other type of tissue? So it's also required in later development.
Yeah, but I think I did check the gene expression pattern. It's expressed in the like the in the other tissues, but not too much, not that high compared to the like the seeds. And I think maybe like because like the seed is lack of the sucrose, and also it's highly expressed in the here. It is highly expressed in the like the outlayer of the endosperm. I was thinking that maybe during the endosperm development there will be some like the bad things happened for the double mutant. That is why we just very hard to get the double mutant.
Okay, so now we do have another question from the chat box, also from Chandler. The question is, what was the permeabilization time you used for your samples? And did you face the leakage of transcripts in your method?
Yes. I really want to like to chat with you, because I just maybe you're also working on that, because I tried for the different times and for the different enzyme. I think definitely not like what we tried 1 minute, 2 minutes, and 5 minutes and 10 minutes. I think it's over 10 minutes, around like 12 minutes but no more than 50 minutes, I think.
Okay. Maybe last question from Wenhao. When you select your telium mutant, did you select based on the durum wheat reference genome or bread wheat reference genome, as the chronos is the durum wheat?
Yes, I just selected with the Chinese spring. Selected with the Chinese spring, because when I analysis the data, I was thinking that because actually I'm not using the Chinese spring. When I try to mapping our samples, I'm using the condenser. So I just using the condenser and to mapping with the Chinese spring. And also when I try to select the telium mutant, it's also the not Chinese spring. It's the chrono anyway. So I was thinking that maybe we just give a try on the chrono, because I think there. I know that the durum wheat already released the genome, and we can compare. But yes, it's a good way to, maybe we should check a little bit. But I think their functions will not change a lot.
Yeah. Okay. It's just a comment from Chandler to say thank you for the answers and also for the talk. I think we'll all learned a lot and enjoyed this talk. Yeah, I think that's the end of this webinar, and we'll see you next time.
See ya.
Okay, bye-bye.
Bye.